Bismillahirrahmanirrahim. We start another practical as asked by many of you to give you more papers on paper 3. So I am now going to be doing a couple of paper 3s for you all to really understand them and be able to do a good job in the coming exams. Now as you can see, this paper is for 2 hours and this will be done in the lab, and you will have to go to the laboratory and do this. Now we will of course get instructions before the exam for the supervisors in the lab to arrange for all the apparatus and all which will be present, which has to be present for the exam. Now there are different variants of it. In Pakistan we do paper 3-3 and paper 3-4. Now of course I'm handling a number of other variants, but they're all the same, they're all the same pattern, they're all the same level of difficulty. So not that you can say that the paper 3-3 was easier and the 3-4 was difficult, because the grade boundary changes for the practical. Maybe a mark or two might be lesser or more, that's it, not much difference there is. So it depends on which paper you're taking. You might be taking the 3-3 in this year, or you might be taking the 3-4, but it's the same thing. Now let's look at question number 1. Now you can first read this as a theory paper, and then that helps you to do this paper even more effectively. I have students who've read this as a theory paper and done this as a theory paper, and they will be getting 38 out of 40 in this paper. And this is a very good score if you can get that in this paper. So now I'll teach you how we do it as a theory paper. Some fruit contains large quantities of ascorbic acid, vitamin C. Ascorbic acid can be absorbed through the partially permeable wall of the gut. You will investigate the rate at which ascorbic acid from 2 gram dm cube ascorbic acid solution diffuses across a partially permeable membrane. So we're going to discuss the rate across a partially permeable membrane. Now, dialysis whisking tubing acts as a partially permeable membrane. You are provided with material shown in table 1.1. You must go through these. A is ascorbic acid, it is 80 cm cube. W is distilled water, is 200 cm cube. D is the dialysis tubing and distilled water, that is 20 cm. The distilled water. The iodine solution is 25 cm cube, is an irritant, and the starch solution is 20 cm cube. So now this will be of course given to you. There will be different beakers and they will be labeled with A, W, D, and I, and S. So you have these different beakers in front of you. Do have a look at them, look at their volumes, think about it. It is recommended that you wear suitable eye protection. Why? Because you might spill something by mistake on your face, and of course that could injure your eyes. You need to allow ascorbic acid to diffuse out of the dialysis tubing into the distal water surrounding the dialysis tubing. So there is this beaker in which you have the dialysis tubing, and the ascorbic acid inside is going to diffuse out, right? So allow ascorbic acid to diffuse out of the dialysis tubing into the distal water, and inside here you have distal water here around this dialysis tubing. Estimate the concentration of ascorbic acid that has diffused out of the dialysis tubing. So I want to find out how much is the ascorbic acid. Say the ascorbic acid was 100 and now inside here it is 20. So that means 20 has diffused out of the 100. So estimate the concentration of ascorbic acid that have diffused. So we want to find out how much ascorbic acid is there in this water which is surrounding the dialysis tubing. Now you carry out steps 1 to step 27. Now I'll walk you through these steps and then you can understand them a little better. Now let's look at step 1. Tie a knot in the dialysis tubing as close as possible to one end so that the end is sealed. Now how do we do this? Now you can see it here. I've given you a picture here. This is how you do the knot. So you tie a knot to the dialysis tubing, and then you can just fill it with anything that you want to fill it in. And this is the sort of diagram that you're going to look at. So pause the video here, have a look at this. I'm sure you've all done this in school. I make my students do this many times. So everybody has a rough idea how this is going to be done. So tie a knot in the dialysis tubing as close as possible to one end so that the end is sealed. To open the other end, wet the dialysis tubing and rub the tubing gently between your fingers. Now, this is a little difficult, but it does happen. You just have to be a little patient and persistent. Patient and persistent. Put 10 centimeter cube of ascorbic acid solution A into the open. So this will be the open end. If you look at this diagram which I'm showing you, this is the tube, this is the open end, and then in this here you put in 10 centimeter cube. So this is going to be filled up with 10 centimeter cube of 10 centimeter cube of ascorbic acid solution A and into the open end of the dialysis tubing. So you take a syringe and you put this in it and you pick it up with a syringe and you put this solution in it. Then step 4, you rinse the outside of the dialysis tubing by taking this and dipping them in here, dipping it in the container of water labeled D. Now carefully place the filled dialysis tubing bag into the large test tube. So you place this here now. Let me just repeat this. Carefully place the filled dialysis tubing bag into the large test tube and secure in position using an elastic band as shown in figure 1.1. You can see this diagram here. This is the elastic band, this is the dialysis tubing, this is the ascorbic acid solution, and this is the knot. Now you will need to fill the large test tube with a known volume of distilled water. So we've got to put some distilled water here in this tube outside the dialysis tubing. So this is what we have to put in something here. Now, so that it just covers the liquid in the dialysis tubing. So it should be something up to here. Now, using a syringe, you use a syringe to add the distilled water to the large test tube and record in A2 the volume added. State the volume of distilled water, but it could be anything between 15 and 30. So I say, well, okay, let me put 20 cm3, but it can be 25, but anything between 15 and 30 was allowed. Because of course you couldn't put in 100 in it because it's a test tube. It's not something you can just put in. Of course it was, we knew what sort of a test tube we had to give you, a boiling tube or whatever. Then start step 7, start timing and leave the dialysis tubing bag in the distilled water for at least 15 minutes. Now, when it says at least 15 minutes, then you know that it can be more than 15 as well. So the question tells you something at least 15 minutes. While you are waiting, continue with this question. You will need to dilute the 2 gram dm cube of ascorbic acid solution A to provide a range of known concentrations. You will need to make 20 centimeter cube of each concentration of ascorbic acid. Some of you still don't know how to do that even though you're in A2, and that is very sad when I look at that. Table 1.2 shows how to make up 2 of the concentrations you should use. Now in this part 2, this is for 2 marks. I mean, everybody should get these 2 marks. There's no question why you shouldn't get these 2 marks. Decide which other concentrations of ascorbic. They had given you this, the first line was given to you. The total volume should have been 20. That was given in the, it was written in the question. It said here, you will need to make 20 cm cube of each concentration of ascorbic acid. Now this is called a simple dilution. This is not called a serial dilution. In another paper, I will discuss with you serial dilution. So this is called simple dilution. And if you have an issue on understanding it, please go on YouTube and look up a video on simple dilution A levels, and you should be able to figure this out. So if you took 0.5 of the 2.0 gram dm cube, then you took 0.5 of that and you added, if you want to make 0.5, actually you wanted to make 0.5 gram dm cube, you added 5 of the volume of ascorbic acid solution, and distilled water you added 15, right? The ascorbic acid was of course the stock solution of the ascorbic acid. As you can see here, the ascorbic acid was here, you can see it here in this table. The ascorbic acid was 2 gram dm cube ascorbic acid solution. This is the stock solution, this is the A. So this was the stock solution, and then you had to make a dilution of it. So you took 5 of that and added 15 of the distilled water to it. Then to make 1.0 gram dm cube, you added 10 of the volume of ascorbic acid solution and 10 distilled water. And then to make 1.5, you added 15 cm cube and you added 5 cm cube of distilled water, and you got a 1.5 gram dm cube. So this was the concentration of ascorbic acid. You know the original stock solution was 20, so you would always make less than 2. So it was 2.0 gram dm cube was the concentration of ascorbic acid of the stock solution. So you would naturally decrease it. You can't increase it. So this was for 2 marks. Then using the beakers provided, now let's look at this part of the question. Using the beakers provided, make up the concentration of ascorbic acid stated in table 1.2. So you make those 3 different concentrations. Put 1 centimeter of SAR solution into a clean test tube. Okay, now let's do a simulation. So this is the test tube, and you add 1 centimeter cube of the star solution. So you add 1 cm cube of the star solution S in a clean test tube, and then you add 5 cm cube of ascorbic acid solution. of the 0 gram, which was the distilled water. So you added 5 cm cube of that, of the zero distilled water. This is distilled water, right? So you added distilled water into the same test tube and shake gently to mix. Now this is an error. Shake gently to mix means how gently? I mean, everybody's gently is slightly different. Maybe you swirl it 3 times. Maybe you swirl it 5 times. Fill a syringe with 2 centimeter of iodine solution. And then you added iodine to it. So you added 2 centimeter, fill a syringe with 2 centimeter of iodine solution. You took a syringe and you filled it with 2 centimeter of iodine solution in it. And you added 1 drop to it, to the mixture of S and ascorbic as a solution. Mix gently. Then you say push gently, syringe resting on top of the test tube, 1 drop of iodine solution, release mixture of S and ascorbic acid solution. Now, do this like I'm doing it with you. Just do this diagram, do this little bit of diagram with a pencil on the paper and explain this to yourself. Then it says you should see a blue color. Now, this is the end point you're looking for when you test the remaining ascorbic acid solution. So immediately it did not have any, so it is going to turn blue. So the blue color is the end point. This blue color is your end point. As soon as you added 1 drop of iodine, no ascorbic acid, it had no ascorbic acid, remember that. It has 1 drop. Mixture of S and ascorbic acid was 0. Add 1 to the S and ascorbic, but it says put 5 centimeter of the 0 gram dm cube ascorbic acid solution, which is distilled water. Now record the volume of the iodine solution you added, whether it is 1 drop or 2 drops, but you have to record it in cm cube, which is the volume. Then you put 1 centimeter of star solution S in a clean test tube, and then you put 5 centimeter of the lowest concentration of the remaining ascorbic acid solution into the same test tube. Now remember it was 0.5, 1.0, 1.5 and 2.0. So this was the lowest concentration, 0.5. So put 5 centimeter of the lowest concentration of the remaining ascorbic acid solutions. You took a test tube, you put 0.5 ascorbic in the same test tube. You put 1 centimeter of starch. So we start off again. 1 centimeter of starch, S, and then 5 centimeter of 0.5 ascorbic acid, right? And then you fill the syringe containing iodine I to the 2 centimeter level again. Add 1 drop of I to the mixture of C. So you keep on adding I to it. The iodine is the I. And ascorbic acid solution as shown in figure 1. Continue adding drops one at a time, mixing after each drop until you see a blue color. As soon as you see a blue color, you wait 10 seconds. If the blue color disappears, then add another drop. Continue adding drops until the mixture stays blue for at least 10 seconds. This is the end point. Record the volume of the iodine solution you've added. Now this of course will be in cm cube that you have added. Repeat steps 15 to step 22 with the other concentrations. The other concentrations were what? 1.0, 1.5 and 2. So you repeat this whole. Now think of this theoretically. Record your results in A3. Record your results in an appropriate table. So whether you do this or you don't do this, you can actually get away with not doing this, and you can do it. So now I'm going to make a table of this result and record your results in an appropriate table and look at this for 5 marks. That is the most important thing. You have to get this 5 out of 5. Now you can see here how I have made the table. Now what are the 5 marks for? The 5 marks of first is the heading. If you wrote this heading, concentration of ascorbic acid in gram dm cube, you got 1 mark. So 1 mark for this heading. You get 1 mark for this heading. You don't do anything, you get 1 mark for this heading. Then you wrote volume of iodine in cm cube. You wrote the units in the heading, you got 1 mark for this. So 2 marks you got without doing anything if you just wrote that and you made the table. Then results for all the concentrations. So you gave me results for all the concentrations. You got a mark for that. Then the greater the volume of iodine for the higher concentration of ascorbic acid. So as the ascorbic acid has increased, more and more of the iodine is needed to convert it. So this was but very simple because it was in the paper that you added one drop and the color changed to iodine when there was no ascorbic acid in it. So if there's more ascorbic acid, it is going to require more and more of it to change the color. So more the ascorbic acid, more the color change. So then records results to appropriate accuracy. I mean, you wrote it to point 0.2, zero point 0.8, 0.8, 1.2, anything. You see, any result would have been okay. Even if you had said, if somebody had said 1.0, 1.5, 1.8, 2.0, 2.4, something like this. Or even if you said 1, 2, 3, 4, 5 cm cube. Well, fine, that was fine, but you have to realize it should have been less than 2 centimeter because they asked you to fill up the iodine to 2 centimeter cube. So there was something in the question which you figured out that it couldn't have been more than 2 cm cube, but you could have also taken more. It also said that if you needed, you could take more of the iodine. So this is the table, and this is the 5 marks for the table. Then it says step 24, remove the dialysis tubing from the large test tube and place it in the beaker labeled for waste. Stop timing and record the time. Now it was less, it was at least 15 minutes. So maybe you left it for 16 minutes or 17 or 18 minutes. But it depends because it says at least 15 minutes, but it didn't say exactly 15 minutes. Maybe when the time you figured it out is 15, it was actually 16 minutes. So stop timing and record the time that the dialysis tubing has been left in the distilled water. Time from step 7 to step 24. So maybe 15, but it had to be something 15 or more than 15. Pour the solution from the large test tube into a small beaker. Use a syringe to remove a 5 centimeter sample of the solution from the small beaker. Then repeat step 15 to step 22 using this sample. Record your results in A4. Record the volume of iodine added. Now this could be anything. This could be anything. So it's not fixed at you, but you could come up with anything. Now record the volume. Say it was 0.8. Now if you look at this one, 0.8 is 0.5. So anything, if it's at 1.0, then 1. But there was no one answer. It was I was going to just check your paper in what you had written. I didn't have to look at the pattern of the whole class or anything. I would check your paper. If I was the examiner, I'd check your paper according to what is given in your table. So if I wrote 0.8 cm cube, then you use your results in A3 and A4 to estimate the concentration of ascorbic acid in the solution outside the dialysis tubing. That was 0.5 gram dm cube. Calculate the rate of diffusion of ascorbic acid. Now that's very simple because how do you calculate rate? Rate is always something to do with time. If you look at my videos, I always talk about that. So it is 0.5 divided by 16. Why? Because it is 16, it was divided by 16 minutes. In 16 minutes, 0.5 had diffused out. So you shows the division by the time, whether it was 15 or 16 or 17, and you give the answer in gram. You give the answer in gram, dm cube, and minutes. So 0.5 over 16, because I said here it's 16 minutes. But if you said 15 minutes, then you divide by 15. If you said 17 minutes, divided by 17. If you said 18 minutes, divided by 18. And whatever your answer was here, it was not the whole question of the whole class. Everybody could have a different answer and it could be correct. And you would get your 1 mark here and your 1 mark here. So now this was how you solved it and you wrote the answer. This is the important thing is you wrote the units and you wrote this whatever the answer was, whichever was the answer, it will be different for every student. Then coming to the part 7 of the question, identify two significant sources of error when finding the concentration of ascorbic acid in the sample from the large test tube. Now it is difficult to read the scale on the syringe. Why? Because the iodine has a color, it's a brownish sort of a color, so you can't read the scale on the syringe. Then lack of suitable precision. Why? Because the volume of the drop is too large. Maybe a little, if a smaller drop would change the color, but we're adding the whole drop. Difficult to judge the end point because the whole drops are added each time. Then imprecise start time depending on when the dialysing tube was added to the distilled water. Then rate of diffusion changes with time. It is highest at the start and decreases as the concentration gradient decreases. Then volume of water added to the outside of the dialysis tubing not precisely determined, so experiment could not be reliably repeated. You just added water to cover it up to a certain point. So all these things were sources of error. Difficult to read the scale on this range, lack of suitable precision, difficult to judge the end point. Whenever there is a color change, that's one of the points that you can write in every paper. Then imprecise start time depending on when the dialysis tubing was added to the distilled water. Then rate of diffusion changes with time. The concentration gradient changes. You see, if initially 100 was inside and only 5 was outside, so the gradient is different. Then it's 90 and it's 10, or it's 80 and it's 20. So the gradient keeps on changing. So rate of diffusion changes with time. Then volume of water added to the outside of the dialysis tubing was not specified. It just said add something to cover it up. That was something which they left in the papers for you all to come up with that idea that there was an error in the experiment. Then coming to part 8 of the question, suggest how you would make two improvements in this investigation. Repeat and find mean. This is a very favorite one which you can write in every paper. Repeat and find the mean. When you repeat the experiment, then you remove the anomalous reading, so it becomes more reliable. Repeat and find the mean. Use of a burette. Instead of saying drops, use a burette. Then use a colorimeter. Whenever there's a color change, then the best thing to use is a colorimeter, which is going to measure the color very accurately. I said very accurately. And use a white card behind the tube. You use a white card behind the tube so that you can know when the blue color has appeared. It becomes more obvious, it becomes more conspicuous. So use a white card behind the tube and find the mass of iodine. Find the mass of iodine added. This of course gives it more accuracy. Now coming to the B part of the question, fruit can be preserved by reducing its water content. This can be done by cutting the fruit into small cubes and soaking the cubes in concentrated sucrose solution. Cubes of melon were soaked in 45% sucrose solution for 20 hours at a temperature of 25 degrees Celsius. The water content of the melon was determined every 4 hours. The results are shown in table 1.3. Table 1.3, time in hours, water content. Now remember this, the first column is always the independent variable. This is what you have changed. You must know what is independent variable. We talk a lot about it in paper 5 as well. And this is going to be plotted on the x-axis. Always, this is the rule. First column F, I, X. First column independent variable is always plotted on the x-axis, and this, the second column or that, is always plotted on the y-axis. So please remember, this is a rule. I don't want anybody changing this and making a mistake and losing a mark in this. Part 1, plot a graph of the data in table 1.3 on the grid in figure 1.3. Use a sharp pencil. That's it. See, you have to write the whole thing. Please, there should be no abbreviations. Now, if you look at it here, this is time in hours. So I have written time in hours here. And then on the y-axis, it is water content slash gram of water for each gram of melon. So water content slash gram of water for each gram of melon. Now just doing this gets you 2 marks. No, gets you 1 mark. The second mark is for the scale. What scale do you choose? Now when choosing the scale, remember what's the smallest number. Here you can see it's 0 and here is 20. Here you see it is 0.20 and it is 1.44. So please divide the scale so that the whole grid is used. Now I have divided it according to this. The time is every 2 centimeters. 2, please write every 2 centimeter, 2, 4, 6, 8. And then on the y-axis it is 0.2, 0.4, 0.6, 0.8, 1.0, 1.2, 1.4 and 1.6. And now you have to plot the points and then you have to join the points. So when you look at the graph, the 4 marks for the graph were labeling the x and the y-axis, then choosing the scale got you another mark if it was correct according to the Cambridge mark scheme. And the third was plotting of the 6 points using a small dot in a circle or a cross. Either you put a dot and you circle it, or you put a cross. So either is going to be a dot and a circle. Please do not circle the cross, or you do crosses and that would be fine. And at least 5 plots joined with a thin line passing through all the points. So when the line, you must pass through all the points. If there's another point here, then it must pass through this point. So this is how you have to do it. Have to plot the graph. Then coming to the last part, explain the change in water content of the melon cubes between 6 hours, between 16 hours and 20 hours. So there was no net movement between 16 and 20 hours. You can see what had happened. The 16 and 20 hours here, you can see it here. It remained 0.20. So there was no net movement of water in or out of the fruit, and there was an equal water potential in and outside. So there was no net movement of water. So this will get you your 2 marks. The word is no net movement because there is movement of water, but there's if 10 molecules of water moving in, 10 move out, so there's no net movement of water in or out of the fruit, and there's the equal water potential. But water keeps on moving. If 10 move in, 10 move out, then there's no net movement of water. So this is exactly how we want you to word it, and it's a very favorite question in this. Now I'll complete the graph for you and let you have a look at it once I've completed the graph. Sorry, on the x-axis you have to take 2 centimeter 4, so it will be 0, 4, 8, 16, and 20 because you had 0 to 20. There's actually a mistake in the mark scheme, and that's wrongly written in the mark scheme. So this was the one that we had to correct. Now you can see I plotted the graph, and these are the plots that I've done. So I plotted the graph. Now what I have to do is I've got to connect them, and I will just connect them and show them to you how that looks like. So connect them with a ruler, point to point with a ruler. That's a much easier way to do it. I'll try if I can do it with you all. So this is going to come here, then this is going to come here, and then it's going to come here, and then it is going to come here. So point to point with a ruler is the best way. Of course, you can do a curve as well, but it must go through all the points. So that finishes your graph. That finishes this part of the question as well. Now you can see this question was for 22 marks. Now out of 22, everybody can at least get 20 out of 22. I'm 100% sure that if you look at it carefully, you understand how this paper is done, you can maybe not even do anything and still get full marks in it, considering that using it, taking it as a theory paper and doing it theoretically. And I'm sure all of you can get very good marks in it. So best of luck and thank you. And please do leave comments, and I will be doing some more papers on the paper 3 so that you all can get a bit of practice in how to handle this paper. Thank you once again.